Zoll LifeVest is a proud sponsor of CardioNerds. Zoll has continuously improved LifeVest for over 20 years, leading to a median real-world wear time of 23.4 hours per day and an AI-enhanced algorithm that delivers zero median false alarms at 90 days. The newest LifeVest garment is lightweight, breathable, and preferred by patients for its comfort. Learn more at lifevestinnovation.com.
Hey CardioNerds, this is Jenna Skowronski, Chair for the CardioNerds Heart Failure Council and currently an Advanced Heart Failure and Transplant Fellow at Vanderbilt. I'm here with CardioNerd Academy Fellow Dr. Apoorva Gangavelli and expert faculty Dr. Ronald Witteles, an Advanced Heart Failure and Transplant Cardiologist from Stanford. Dr. Witteles has particular expertise in the treatment of amyloidosis, serving as the co-director of Stanford Amyloid Center. Today, we'll be taking advantage of that expertise to discuss the Nex-Z trial, a phase 1 trial assessing the safety profile of the drug Nex-Z that utilizes CRISPR gene editing technology in ATTR cardiomyopathy. This trial was presented at AHA 24. Let's get started with a case. Apoorva, take it away.
Hi, Jenna. It's great to be here. Today, we have a 78-year-old male diagnosed with wild-type ATTR cardiomyopathy, presenting with recent symptoms of reduced exercise tolerance, leg swelling, and orthopnea. He's been hospitalized multiple times in the last year for acute decompensated heart failure. His home medication regimen includes carvedilol, lisinopril, spironolactone, and furosemide. He was first diagnosed with heart failure 2 years ago. He currently has an NYHA Class 3 symptoms. He presents to the CardioNerds Heart Failure Clinic today wondering about treatment options for ATTR.
Dr. Witteles, aside from the Nex-Z trial, what do we know about the management of this patient? What are some recent advances we've seen in treatment options for ATTR?
Well, thanks, Jenna and Apoorva. This is an amazing time in ATTR amyloidosis. I kind of can't believe how far we've come when you think about where we were a decade ago with no treatment options, the only way to diagnose the disease being invasive endomyocardial biopsy. And now we recognize it's really not that rare, particularly in older populations with heart failure. It's easy to diagnose. There's some pitfalls, but overall it's easy to diagnose. And we now have multiple treatment options, FDA approved, and many more in development, including, of course, Nex-Z, which is the main topic we're talking about tonight. So for this patient, I always like to think of it in a couple of different ways. One is how can we best manage the patient independent of the amyloidosis-specific therapies? And then two is what can we do to actually affect the progression of this patient's amyloidosis? So for the former, well, let's take a look at it. So he's on carvedilol, lisinopril, spironolactone, and furosemide. Well, clearly the diuretics are likely to be needed as described. And of course, that's a matter of finding the right dosing. Often these patients are under-diuresed, but presumably the CardioNerds Heart Failure Clinic knows how to manage loop diuretics. A MRA like spironolactone pretty clearly would be helpful in this situation as well. The drug that jumps out from a heart failure standpoint that this patient is missing is an SGLT2 inhibitor. While there is no randomized trial evidence in cardiac amyloidosis for SGLT2 inhibitors, of course there is for both HFpEF and HFrEF. And there's quite a bit now of observational evidence as well as clinical experience to suggest that this is a class that really benefits patients with ATTR amyloid cardiomyopathy. So I would like to add that. Then we look at his carvedilol and lisinopril. So classically speaking, we talk about standard GDMT, at least when it comes to RAS inhibitors and beta blockers, as being contraindicated in patients with cardiac amyloidosis. That's probably a bit of an oversimplification. A lot of these patients with wild-type ATTR cardiomyopathy are elderly patients with hypertension, and to just pull all of it away and let them be frankly hypertensive is clearly wrong as well. But don't feel like you have to push those agents as you would in a classic, at least HFrEF patient. And then with beta blockers, as we know, most restrictive cardiomyopathy patients don't do particularly well with beta blockers. And I would say, unless it was needed for rate control of arrhythmias, you would want to scale that back or pull it off completely. Then the next question is, how about arrhythmias? And so we didn't hear about it, but if he was in atrial fibrillation, we'd want to address that. Is he having problems with heart block, which again plagues these patients quite frequently? Okay, we've gotten past that. Now let's get to amyloid-specific therapy. So as I was mentioning, this is a really exciting time. We've, of course, had tafamidis available now since really 2019. It's been commercially available and has a massive effect, massive treatment effect in terms of improving overall outcomes. Though interestingly and differently than most therapies in heart failure, it has the largest treatment effect in patients who are diagnosed earlier. So while you would clearly use it, you would worry that this is a patient who it sounds like already has pretty advanced disease, and you may not get as much benefit from it as you would in a patient who's earlier. We recently had the FDA approval of the second agent, acoramidis, which, as the name implies, is similar to tafamidis in that it stabilizes the circulating transthyretin protein. And it clearly also has a massive treatment effect. Really impressively dropped cardiovascular hospitalizations in a large randomized controlled trial by 50%, which is, again, just a massive treatment effect, far higher than we think of for most classic heart failure therapies for either HFpEF or HFrEF. Is it better than tafamidis? We don't know for sure because they weren't checked head to head, but it is clearly an effective therapy and in a test tube, probably a bit more potent as a stabilizer. Then we have another series of drugs that is clearly on the way of the silencers. These are knocking down the liver's production of the transthyretin protein. And the one that we can say with some confidence is likely to get approved in early 2025 is vutrisiran. And that was from the Helios B study, which was another randomized placebo-controlled clinical trial. Once again, showing a massive treatment effect in terms of mortality, in terms of hospitalizations, and in terms of the outcomes such as KCCQ and 6-minute walk. So there's a lot that you could think about. Right now, the only approved options for him would be either tafamidis or acoramidis, and you can make a case for either one, but you would certainly want to start one of them. And then finally, of course, you'd want to think about clinical trial options, which might include Nex-Z.
Thank you so much. That's great background. Going into the details of the Nex-Z trial, this is a phase 1 single-group open-label study, which included 36 patients with ATTR. Key inclusion criteria included ages between 18 to 90 with a diagnosis of variant OR wild-type ATTR, at least one previous hospitalization for heart failure, NYHA classes 1 to 3 symptoms, and an NT-proBNP greater than 600. There was both a dose-finding portion of the study and then an expansion portion, with a fixed dose in both. Patients received a single dose of Nex-Z over 2 hours with a pretreatment of steroid and antihistamine to prevent infusion reactions. The primary objectives of the study were to evaluate the safety, side effect profile, and pharmacodynamics of Nex-Z. The serum TTR level was the primary measure of pharmacodynamic activity. Secondary endpoints were changes in the levels of NT-proBNP and high-sensitivity cardiac troponin, along with changes in the NYHA class and the patient's quality of life on the Kansas City Cardiomyopathy Questionnaire, echocardiography, and CMR imaging changes, as well as a 6-minute walk distance and in cardiopulmonary exercise testing.
Amazing. Thanks, Apoorva. Before we get into the study's findings, let's pause for a moment to discuss CRISPR-Cas9 technology. This is a gene editing technology that won the Nobel Prize for Chemistry in 2020, and as a chemistry major, I am a huge CardioNerds fan of this. So it actually creates targeted permanent modification of DNA in an organism, humans in this case, by adapting naturally occurring genome editing systems found in bacteria. CRISPR allows for the identification of a target gene, and Cas9 is an enzyme that cuts DNA. In combination, you can target and permanently edit genes. This technology has caused a lot of excitement in the medical field for its potential in disease treatment, as well as some questions about long-term implications. Dr. Witteles, what has been the conversation in the amyloid community around CRISPR?
Well, I'd say it's a lot of excitement. When you think about CRISPR, in many ways, ATTR amyloidosis is the perfect disease to target, or at least one of the truly perfect diseases to target, which is why, of course, it's one of the first to have clinical trials in the space. Why is it perfect to target? Because it's a single protein disease. There's not that many, certainly in the adult population, that's the case. And number two, we know from the silencer trials that I was just referencing that if you knock down production of the protein, that good things happen. And unlike, let's say, SGLT2 inhibitors, which we know work in heart failure, but we don't really know why. We know exactly why tafamidis works, we know exactly why acoramidis works, and we know exactly why the silencers work. They were designed to do exactly what they do. In the case of the silencers, it's to knock down TTR production. So if it works to knock down TTR production by one mechanism, it should work as long as you can safely and effectively knock down TTR with another mechanism. The other thing that I think sometimes causes confusion when you think about gene editing with CRISPR in ATTR amyloidosis is that people think of it as, oh, we're going to edit the abnormal gene, a variant or mutant gene, to the wild type, which is not what's being done. So instead, this is editing it to just knock down production, period. And as such, it could be used for either the wild type or the variant form of the disease. And then the thing that is so attractive to not only providers, but maybe even more so patients, is that you could achieve this theoretically with a single one-time IV therapy, and it should knock down the levels forevermore for the patient. So though the safety, of course, needs to be demonstrated, the efficacy, even though I would be shocked if it wasn't efficacious, the efficacy obviously still needs to be demonstrated. It's for these reasons there's a lot of excitement.
Thank you so much. That was really helpful. And with that primer on CRISPR technology, let's get into the trial results. So the patient population, they enrolled 36 patients, 31% of whom had variant ATTR cardiomyopathy with 1 of 5 pathogenic TTR variants. The median NT-proBNP was about 2,050, and 50% of these patients were NYHA class 3. And then the median follow-up was 18 months. As a reminder, this is a phase 1 trial, so this is largely focused on safety. There was at least one adverse event in 34 of the 36 patients. 5 patients had transient infusion reactions, and 2 had mild transient elevations in liver enzymes. 14 patients had serious events, though most of these were felt to be consistent with their underlying ATTR cardiomyopathy, including heart failure and arrhythmia, rather than from the drug. Patients were noted to have URIs, COVID, and UTIs during the study period. For TTR levels, the data has caused a lot of excitement around serum TTR protein levels, which dropped with a rapid and seemingly durable response. The mean percent change was 89% at 28 days and 90% at 12 months. TTR level reduction was sustained at 24 months in 11 of the patients who finished a 2-year follow-up. Secondary outcomes were measured, but as a reminder, this is a phase 1 trial, so these will be tested again kind of going forward in the phase 3 trial. But troponin and NT-proBNP were stable during the period. Median change in 6-minute walk distance was an increase in 5 meters. Median change in overall KCCQ scores was 8 points, with 61% of patients having at least a 5-point increase, and 47% of the patients had an improvement of at least one NYHA class. Measurement of cardiac remodeling at 12 months with either echo or CMR showed stability. So, Dr. Witteles, this is a phase 1 trial, but where do you think this technology is headed in terms of clinical application? What implications do the mechanism of action have on what we would need to intervene on the natural history of ATTR cardiomyopathy?
So, again, I think that the results here are really pretty spectacular. One IV infusion, and forevermore, you see 90% knockdown in production of this protein. That's incredible. And I say forevermore, it was obviously only looked out to a couple of years. By the way the drug works, there's no reason to think that there would be a rebound suddenly 2 years later. So it is almost certain that this will be sustained. And it was consistent. It wasn't like some patients had sustained and others were having rebound. Pretty clearly, you knock it down by roughly 90% in everybody who gets this drug. Now, coming back to what I was saying before, the TTR protein, whether it's going to deposit or not, there's no reason why it should care the mechanism that led to it being produced less. Right? At the end of the day, you have a certain amount of circulating TTR protein, and the less that you have, the less is going to deposit. And so we know that really good things happen with 80% knockdown on average with vutrisiran, and we know that definitively from the Helios-B trial. I just find it impossible to believe that 90% knockdown wouldn't be at least as good, and we'd have every reason to think that it might be better. In AL amyloidosis, the more you knock down the precursor, in this case, the clonal light chain, the better results you get. We don't know that the exact same thing will be true in TTR, but we can say with confidence, there's no reason why 90% knockdown shouldn't be at least as good as 80%. So in my mind, though, of course, you have to prove it in a clinical trial to get FDA or EMA approval, I have little doubt that the drug will work. It's just a matter of proving it and thinking about the right clinical trial design. Safety, as you highlighted, is maybe the thing to think about more, because this is a still new technology, and we have very safe alternative agents that we can use. Tafamidis, acoramidis, vutrisiran, all a totally safe, clean safety profile. So when we think about this, it's got to be safe. Overall, the results here were really encouraging. I think the thing that most of us looked at most closely was, were there going to be hepatotoxicity just because of where this drug is acting, because of where TTR is being produced, which is the liver? And overall, that looked quite good. There were 2 patients who had very modest elevations in ALT, and it was transient and it came down. And as you noted, a few patients had infusion reactions, but nothing that ended up having any sustained problems. Now, look, this is not a trial of hundreds of patients. It's about 36 patients. So clearly safety is still going to be an important thing to look at in the phase 3 trial, but overall really encouraging. So when would we need to intervene in the natural history of ATTR cardiomyopathy? As early as we can. A consistent thing that has been seen across all of the phase 3 trials to date is not just that outcomes are better when you treat early. Of course, that's the case. Among other reasons for lead time bias, it's always going to be better if you intervene early. But you actually seem to have a bigger treatment effect when you intervene early. So we need to, in the disease, continue to push on awareness and moving the diagnosis up earlier. And it's not the sort of thing where you would want to wait and say, well, let them develop worsening heart failure and then we'll intervene.
Dr. Witteles, what are some limitations you noticed when reading this trial?
Well, the limitations are that it is a phase 1 trial. I mean, it's what it really comes down to. So I think safety, we talked about positive, but obviously going to need to be seen in bigger numbers. Efficacy, if we talk about it for knocking down TTR, I'm sold. That clearly works consistently. But when you're talking about those clinical endpoints, the changes in biomarkers, the changes in imaging, small numbers, some of them like KCCQ or 6-minute walk can be affected by patient effort or bias in a non-randomized, non-blinded trial. So you want to be careful saying, oh, well, clearly when you compare the effects that it had here on NT-BNP versus some of the phase 3 trials, it looked even better. Well, yeah, that's true, but that's comparing apples to oranges. So I think the main limitation is to say, look at the TTR knockdown, look at the safety, but look with a grain of salt at the so-called clinical outcomes just because of the numbers in the study.
So what's in store for future investigation regarding Nex-Z in the treatment of ATTR?
Well, so based on this positive data, and by the way, just to show you the level of excitement, this New England Journal article was the second New England Journal of Medicine article from the same phase 1 trial, which may be a record. So tells you how much excitement there is for both this technology and this field. But anyway, based on this sort of very encouraging data, a phase 3 trial was launched. It is currently ongoing. We are currently enrolling in it at our site. And this is a definitive phase 3 planned 765 patient double-blind randomized placebo-controlled trial, randomized 2-to-1. So 2/3 of the patient would get Nex-Z and 1/3 would get placebo, with hard primary endpoint of CV mortality and CV events, which is mainly, when we say CV events, we're mainly talking about hospitalization. So very similar in design to the other phase 3 trials, and is the sort of obviously definitive trial that would be needed to ultimately lead to regulatory approval. There is also a similar study in a smaller number of patients, because it's a much rarer disease, that is planned for patients with ATTR polyneuropathy, which only occurs in the variant or hereditary form of the disease. That data will be coming as well. But for us CardioNerds, probably the one that is the main one causing excitement is the cardiomyopathy trial, which is also the much larger one, and the name of it is Magnitude.
Dr. Witteles, we'll have to have you back to talk about Magnitude when that gets published, so I look forward to it. But thank you so much for joining us today and sharing your expertise on ATTR and the insights on the Nex-Z trial. We really appreciate it. Thank you for teaching us. Thank you for your time.
Thank you. Always a pleasure.
Hey everyone, do you dread doing your notes every day? Do you feel like you can never get ahead of charting? If you're shaking your head yes, then keep on listening, because Freed AI is a medical scribe on a mission to free clinicians everywhere. Freed AI listens, transcribes, and writes medical documentation for you. So just imagine leaving your office at the same time as your last patient. Now, wouldn't that be delightful? Freed AI is HIPAA compliant, secure, and takes less than 30 seconds to learn. Team, artificial intelligence cannot replace you, but it can do the administrative work that will allow you to spend more quality time with your patients and enhance your personal work-life harmony. So get back to doing what you love, helping your patients, and let Freed AI do the rest. Check out www.getfreed.ai to learn more.